Hi there, welcome to Spec Point 2.9, which is all about protein structure. Now, this is a big section because it goes all the way from the structure of an amino acid into how the amino acids join together and then into how we fold into proteins. So it's a big spec point, and let's start off with the structure of an amino acid. Okay, so you might have to draw an amino acid in an exam or finish off a diagram of an amino acid. Sometimes you might have to label the parts of an amino acid. Okay, so let's start off with amino acids. So, an amino acid has this structure. In the middle, it has a carbon. At the top, it has a hydrogen. At the bottom, it has an R group, which is different in each amino acid. There's actually 20 different amino acids, so there's 20 different R groups. To the side on this side here, we've got CO, it's a double bond to the O, OH. So you could write that COOH, and that's called a carboxylic acid. Group, or sometimes people just call it a carboxyl group. Right. So sometimes they might give you the structure of an amino acid and ask you to label it, or sometimes they might ask you to draw the carboxylic acid or the carboxyl group on that right-hand side there. On the left-hand side, we've got NH, two H's, so it's NH2. And that NH2 group is called an amine group, or sometimes you might hear it called an amino group. Okay, and that tells you then why it's called an amino acid, because we've got an amino group on this side and an acid on this side, so it makes an amino acid. So make sure you're able to draw that and label what the different parts are. This R group here we've talked about a little bit is called the residual group. Now, obviously, the carbons, the oxygens, the hydrogens are actual elements from the periodic table. The R is not an element from the periodic table. It's just representing what's on various different amino acids. There's 20 different amino acids, so this R represents 20 different structures that you don't actually need to know. Okay. The next part of this spec point is how do two of these amino acids join together? So two amino acids joined together is called a dipeptide, di for two. So a dipeptide is two amino acids joined together, and you'll need to know how this happens and be able to draw it. You're unlikely to have to draw it all from scratch. You'll probably have to finish something that they've started drawing. So remember the structure from here. So you've got your carbon in the middle, H at the top, R underneath. So carbon, H at the top, R underneath. Then on the left-hand side, we've got our NH2, and on the right-hand side, we've got our COOH. So the left-hand side, NH2, and on the right-hand side, C, double bond to the O, single bond, OH. And let's draw another one. So it's carbon in the middle, hydrogen above, R group below, NH2 on this side, C double OH on this side, double bond to the O, OH. Okay, so sometimes in an exam they will draw that for you and they'll ask you to circle the part that's involved in making the peptide bond. So a peptide bond makes a molecule of water. So that's H2O. So the parts involved in making the bond are these just here. All right, which makes a molecule of water. H2, two H's and an O. All right. Sometimes they might ask you to draw the dipeptide that it forms. So let's go again. Carbon in the middle, hydrogen above, R group below, NH2 to the left, COOH, remember the double bond there, to the right. Same again here. Carbon in the middle, hydrogen above, R group below, NH2 to the left, Keep practicing, keep drawing this out. COOH to the right. So if the OH and the OH from just there are involved in making the bond, we can rub those out. And where have they gone? They've gone into making a molecule of water. So we can say plus H2O. Okay. The way this bond then forms, if we just rub out those ones, is we just draw the bond. I'll draw it in a different colour so it's clear. We just draw it usually as a straight line from that carbon across to the nitrogen. Now, you wouldn't get penalised for this next bit, but usually this oxygen, rather than going off to the diagonal now, we would usually, by convention, draw it going straight up. And the same with this hydrogen. So rather than it going off diagonally just there, when it's a dipeptide, the convention is to draw it straight up. Okay, so that now is our dipeptide. Okay, and this bond here is a peptide bond. P for protein, P for peptide. And the type of reaction that's formed that has made a molecule of water. Therefore, that reaction is a condensation reaction. Okay, so going from those two amino acids to this dipeptide, that's a condensation reaction because it's made a molecule of water. Okay, if you wanted to break that peptide bond to make two amino acids again, going that way, that would obviously be a hydrolysis reaction. All right, so let's just label these. So we started off with two amino acids. So two amino acids joined together in a condensation reaction to make a dipeptide and water. Another common question is what are the products formed when two amino acids join together? And you need to say a dipeptide and water. A lot of people forget to say the water, they just say it makes a dipeptide. So just be ready for that question. Don't let that catch you out. The next molecule that then forms once we've joined two amino acids together to make a dipeptide is a polypeptide. So the word poly means many, so this is now many amino acids joined together in a chain. So the same thing will happen again, but it will just happen in a longer chain. Okay, so if we just said that's one amino acid rather than joining it together, drawing it all out, then we've got a peptide bond to the next amino acid. Peptide bond. Let's label these bonds. Peptide bond to another amino acid. Peptide bond to another amino acid. And each of those are going to look like this. Okay, these are the peptide bonds. So that continues, adding lots of amino acids in a chain, and that chain is called a polypeptide. Each individual amino acid is called a monomer. Mono means one. And then when we join them together, polypeptide is called a polymer. Okay, poly means many. So the single unit, the monomer, is the amino acid. And the polypeptide is the chain of them joined together. Many of them is the polymer. Okay, so just remember that. Now, the difference between a polypeptide and a protein is that the polypeptide is just a long line of amino acids, whereas a protein is a specific shape. It's when the polypeptide has been folded up. Now, to make a protein, we've got 4 levels of protein structure that you need to know: primary, secondary, tertiary, quaternary. So let's start off with the primary structure. Now, the primary structure, very simply, is literally just the order of amino acids, and they're in a very specific sequence. Okay, so the sequence of amino acids in the polypeptide chain. And it's very, very specific. Okay, it's not random. So if we were to draw this I drew it a minute ago as just rectangles, but more often than not, they draw them as different shapes. Now, but that's because there's actually 20 different amino acids, and this just starts to represent that there's different amino acids. That they're not all the same monomer joined together, they're actually all different. So that specific sequence of amino acids is your primary structure. At this point, it's also worth always mentioning that they're joined together by peptide bonds. There's often marks for the type of bonds. Okay, so we can say that they're joined by peptide bonds. All right. The next structure of proteins is the secondary structure. So the secondary structure is what we call localized folding. So it's not the folding of the whole molecule giving the overall 3D shape. It's just little bits of folding. And we've got two names for them. The first one is an alpha helix. The second one is a beta pleated sheet. Okay, sometimes you'll see the alpha drawn as the symbol like this. Sometimes you'll see the beta drawn as a symbol like that. It's intentionally got this long tail here. So that long chain of amino acids here, this bit here might be folded into a little helix. This bit here might be a beta pleated sheet. And the reality is that the molecule's really long. So we've got bits that are forming this sort of spiral, alpha helix. We've got some bits that are forming this zigzag, and then it goes around the corner, zigzag back, around the corner, zigzag back. But in the context of looking at the whole molecule, we've actually got some parts of each. So if we were to draw the whole structure, if it might have a little bit of alpha helix, then it might have another bit that's beta pleated sheet, and it might have a bit that's not got either. They might have another bit of helix and another bit of beta pleated sheet, etc. Okay, so we've got this really long polypeptide that's got areas of localized folding. So there's a little bit of folding there, a little bit of folding there, a little bit of folding there. And the two examples of that localized folding are alpha helix and beta pleated sheet. As I said before, we always need to mention the bonds. So the bonds that hold these, both the alpha helix and the beta sheet, are hydrogen bonds. Okay, so let's just add in on here. Held by hydrogen bonds. Okay, which individually are weak, but with a lot of them, they can kind of group together to be strong. Okay, in a lot of books they go into the detail of where and how and what these bonds are, but actually the reality is we just need to know these overall structures and that they are the bonds, but they'd be kind of in here holding these structures together. Right, something like that, and then they could go in between the strands, something like that. All right, as long as you know there's hydrogen bonds, that's what's holding them in place, that's fab. The next stage is the tertiary structure. So that's another word for the third structure. And the tertiary structure is the overall 3D shape. Okay, not localized folding, not localized bits of shapes, but the overall 3D shape. And it's very specific to each type of protein. And the way that we get this specific folding to be different in each type of protein is because each protein has a different amino acid sequence in the primary structure. So this is a really, really common question. So the primary structure, which is the sequence of amino acids, determines this 3D shape because each amino acid has a different R group. So remember we said that there's 20 different amino acids and each of those has a different R group. Now, each of those R groups has different properties and therefore can form different bonds. So the primary structure, the sequence of amino acids, determines the 3D shape because each amino acid has a different R group which has different properties and can form different bonds. And that's what determines how it folds. So this determines how it folds and the shape it forms. So let's have a look at some of these bonds and what's going on. So we know from the secondary structure that we've got this kind of bits of alpha helix, bit of beta-plated sheet. So it's going to fold up in a really, really specific way. So let's just, obviously my picture's a bit random, but in reality it's very specific. So something like that. So this is our overall 3D shape, and let's think about how and why it might form a specific shape. So first of all, the first type of bond is an ionic bond. Okay, so remember we always have to talk about the types of bonds at each stage of protein folding. So the first type of bond is an ionic bond. Now, an ionic bond forms between R groups that have either positively charged or amino acids that have positively charged R groups and amino acids that have negatively charged R groups. So let's say, for example, we had an amino acid here that had a positive charge, one here that had a negative charge. They'd be attracted together and form a ionic bond, and that would pull that bit of the chain and that bit of the chain towards each other. Okay, so rather than these bits folding in a way that are really far apart, these two parts of the chain would fold in a way that pulls them close together. Okay, the second type of interaction that we get is called a disulfide bond. So di means two, sulfide, sulfur. So this happens between S, sulfur, and S, sulfur. So let's say, for example, we had one that had a sulfur there, one that had sulfur on the R group there. They'd be attracted together, and that bit of the polypeptide chain and that bit of the polypeptide chain would be pulled together to form this disulfide bond. Now, of the 20 different amino acids that we've got, only one amino acid has sulfur in it, and that amino acid is cysteine. So we know that this amino acid here and this one here must both be cysteine because they're the only ones that have sulfur. And then we get our disulfide bond. The next one we get, as we've come across before, is the hydrogen bond. And hydrogen bonds form between I can't talk. Between the R groups that have slightly positive and slightly negative properties. Okay, so let's say we had, these are the ones obviously that we had in the secondary structure as well, but let's say we've got a bit here that's slightly positive, an R group here that's slightly negative. They'll form a bond, binding together here. Hydrogen bonds, remember, are very weak bonds, but in quantity they can add up to make strong interactions. And the final thing is hydrophilic and hydrophobic. Now, hydrophilic means water hating, water loving. Water hydrophobic means water hating, like phobic, like scared of. Okay, so we got hydrophilic, which is water loving. Now, our groups that love water, if I just put a star, oops, loving, water loving. If we mark these ones with a star, they're going to always go to the edge of the molecule. So our groups that are hydrophilic are going to be moving to the edge of the protein where there's water. And then our groups that are the opposite have hydrophobic properties, which means water hating. They're going to move as far away from water as possible. So if I draw these as little squares, these are going to move into the middle. So wherever there's R groups that are hydrophobic, they move to the middle of the protein. Okay, and with all of that going on, this polypeptide chain ends up in this very, very specific 3D structure. Because proteins need to be specific shapes to do their job, don't they? So if that protein was an enzyme, it needs to fold in a way that it's got an active site that's a very specific shape for the substrate to bind, for example. If it's a receptor protein, it needs to fold up in a shape that's got a part that's really, really specific shape for the molecule that binds the receptor to bind and nothing else. So it needs to have this really, really specific 3D shape. The final level of protein structure is the fourth level, which is called the quaternary structure. And the quaternary structure actually isn't always present. Only some proteins have the quaternary structure. And it's when a protein is made of more than one polypeptide chain. Okay, so if I draw different polypeptides in different colours, could be that it's made of this polypeptide. See my specific shape? Maybe it's got two of those, and maybe it's got two of these. So you can see that that protein there has a quaternary structure. It actually has four polypeptides. So this is just an example. This one's made of four polypeptides to form one protein. All right, so that's one protein made of four polypeptides. And an example of that is hemoglobin, the oxygen carrying molecule that we get in our red blood cells. That's an example of a protein that's got a quaternary structure with four subunits. Okay. The final part of this spec point is the difference between globular proteins and fibrous proteins. So once we've got our completed proteins, they then fit into these two categories. So oops, right in there. So we've got globular proteins and we've got, I'll just do it to the side as a comparison. We've got fibrous proteins. Okay, so globular proteins, let's think about those first of all. So globular proteins are kind of like a ball, kind of like a sphere, a kind of circular football-like protein. Okay, so they're sphere-like, like a ball. Okay, that kind of shape. They'll have the hydrophobic parts in the middle, hydrophilic parts towards the edges, and this means that they tend to be water soluble. All right, soluble in water. Um, Things like enzymes are the most famous example. And the other example that they specifically want you to know in your course is hemoglobin. Okay, so they're kind of circular, ball-like structures. They dissolve in water, water-soluble, and famous examples are enzymes, all our enzymes. And the specific example that we need to make sure you know is hemoglobin. Um, because they're water soluble, that means that they can dissolve in something like blood and therefore can be transported around the body. Okay, so they're useful for transport because they dissolve in liquids and then can be transported around. And you need to know specifically a few facts about hemoglobin. So hemoglobin is the molecule that carries oxygen in red blood cells. As we said a minute ago, it's made of four subunits. It has a quaternary structure made of four subunits, and the names of those subunits are two alpha subunits and two beta subunits. So there's two of the same, like the drawing that we did a minute ago. So it's got two of one kind, alpha, and two of another kind called beta. Make sure you're aware that it is soluble, like most globular proteins, and that's useful because it dissolves in can dissolve in the blood or other aqueous solutions. Now, the hemoglobin has within it heme groups. Okay, so it contains heme groups and they contain iron. And it's actually that iron that carries the oxygen, that binds to the oxygen, which is why it's so important that we always have iron in our diet. Okay, because we need iron for our heme groups, for our hemoglobin, for our red blood cells to carry oxygen around our body. And it's this iron that binds to the oxygen molecules to carry oxygen around our bodies in our blood. Now, the other type of protein is a fibrous protein. Now, rather than being a long, thin shape, a sphere-like ball shape, it's a long, thin shape. Okay, so if we just draw both of these structures, just to make it really visual, so I'm going to draw them in a very simple way. So your globular protein is round, and your fibrous protein is your long, thin protein. Okay, so we've got these two really distinct structures, circular like a ball or long thin shape. All right, so a little bit more about fibrous proteins that we need to know. So obviously they are not going to be water soluble, okay, because they're so long and thin, there's no way they can hide amongst water molecules, so they're not soluble, we should say insoluble. Okay, the most common example, the example that you really need to know, is the example of collagen. And we'll come on to the details about collagen in just a second. Um, So what is collagen, first of all? Now, it's actually the most abundant protein, the most common protein that we've got in our body, and you find it in bones, muscles, skin, tendons, things like that. And basically, it holds different things together. It forms a sort of scaffold to provide strength and structure in our bodies. We wouldn't want them to be soluble. So you wouldn't want your collagen to dissolve in your blood and move around your body. You want it to stay where it's meant to be. So no, it's not soluble, and we don't want it to be soluble. But it is strong, okay? So it has got the property of strength. All right, and that is important because it needs to be strong because it's a structural protein. A little bit about the structure of collagen specifically is that it's sort of like a twisted spiral. So let's put collagen, a twisted spiral shape. Okay, so I'll just draw you the collagen. So it's a little bit, it's a little bit like DNA, but DNA is a double helix and collagen is actually a triple helix. So I'm not very good at drawing it, but there's three spirals. Terrible at drawing actually. There's three spirals wrapping around each other. Okay, so we call that a triple spiral, and that's held in place by hydrogen bonds. All right, and you just need to know those few details about it. So, like you needed to know about the hemoglobin, that was 4 subunits to alpha 2 beta. In terms of collagen, you need to know that it's a triple helix, which means it's made of 3 helices wound around each other, and that they're held by hydrogen bonds. It makes a very strong molecule that's insoluble. Okay, so just make sure you know those few facts about specifically hemoglobin and specifically collagen because they're the two that are quoted in your specification. All right, and that's the end of that one.